Good morning, AP Biology students. My name is Kevin Sanchez.
And I'm Bryan Portillo, and today we're reviewing the 2024 AP Biology exam long FRQ number 1.
Let's get into it.
First question on the FRQ portion of the exam is asking about the cell life cycle, meiosis, and DNA replication. Number 1 says, crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the first cell division. A1 is asking to describe the function of S phase in interphase. The S phase is a crucial part of the cell cycle, and it is occurring between G1 and G2. It's when DNA replication happens, preparing the cell for mitosis.
They gave us some paragraphs and a diagram. Always make sure to read these paragraphs very thoroughly and examine these diagrams well. They're very important to the questions, and just take anything they give you on the test. So I'm going to read it just for the sake of reading, for better understanding. So some regions of a chromosome called hotspots display a higher frequency of crossing over than other regions do. Crossing over is suppressed in chromosomal regions near the centromeres. The centromere region of a duplicated chromosome includes a collection of proteins that form a structure called the kinetochore. Scientists hypothesize that one or more of these kinetochore proteins are responsible for suppressing crossing over around the centromere. To investigate their hypothesis, scientists modified chromosome 8 in yeast such that in each cell, one chromosome from the pair of homologous chromosome 8s contained the gene encoding red fluorescent protein, RFP, while the other chromosome from the pair contained the gene encoding green fluorescent protein, GFP. Cells expressing RFP emit or give off red light, and cells expressing GFP emit green light. Models of the modified chromosome 8 both before and after crossing are shown in figure 1. So looking at the diagram, A in figure 1 shows before crossing over, and you see here that the centromere is here. It's really just where the sister chromatids link together. And right here we are showing the gene that's encoding RFP, the red light emitting protein, while this one is encoding GFP, the green light encoding protein. So right here we see the hotspot between the two genes. Alright, so part B of figure 1 shows the two chromosomes after crossing over, and as we see here, this chromosome only has one gene encoding RFP, while this one has 3 genes, 2 encoding GFP and 1 encoding RFP. So A2 asks us, explain why some haploid cells formed after meiosis in this experiment will have only one fluorescent marker. And what it's asking us is, why do we have this chromosome here after crossing over when here both chromosomes had 2 genes encoding for RFP? So a good answer to this would be, during meiosis, homologous chromosomes separate. If only one chromosome had the marker originally, half of the resulting cells will receive it.
So moving on to question B, the paragraph says, the scientists then investigated whether attaching individual kinetochore proteins to a specific DNA sequence present in a known crossing over hotspot on chromosome 8 affected the frequency of crossing over at this location. In their first experiment, they examined 3 groups of yeast cells containing the modified chromosome 8. Group 1 contained the kinetochore protein attached to the hotspot. Group 2 contained the kinetochore protein CTF attached to the hotspot, and Group 3 contained the kinetochore protein IML attached to the hotspot. For each group, the scientists determined the frequency of crossing over between the RFP and the GFP genes. To determine the frequency, the scientists added the number of cells emitting both red and green lights to the number of cells that emitted no light and divided by the total number of cells. B1 is asking to identify the control group for the scientist's first experiment shown in figure 2. The control group is group number 1 with no kinetochore added. This group serves as a control because the addition of kinetochore proteins to the yeast would be considered a modification that would result in a modified outcome of frequency, while no kinetochore protein added will result in a natural frequency of crossing over.
B2 of the first question says, in a follow-up experiment, the scientists created a modified version of CTF in which the DNA binding portion had been removed. They compared the frequency of crossing over in yeast cells in the presence and absence of unmodified CTF with that in yeast cells in the presence and absence of modified CTF protein. The data was not shown. In the follow-up experiment, justify why the scientists used a modified CTF protein that is unable to bind to DNA as a control. So this here tells us that they use in a follow-up experiment, they use two different CTF proteins, one that can bind to DNA and one that cannot. And it's asking us why the scientists use a modified CTF protein as a control. So a good answer would be that the scientists used the modified CTF protein that is unable to bind to DNA as a control to ensure that any changes in the crossing over frequency are solely due to the DNA binding ability of the protein.
Moving on to question B3, it is asking to identify the independent variable in the follow-up experiment. The independent variable in the follow-up experiment is the version of the CTF protein before it is either modified or remained unchanged. Because the CTF protein before being modified or unmodified is the same between the two that are compared in the follow-up, making the CTF protein before any changes the independent variable in the experiment.
C says, based on figure 2, describe the effect on the frequency of crossing over when CTF is attached to the chromosome 8 hotspot compared with the effect when IML is attached to the hotspot. So if we look at figure 2, we see that when CTF is attached to the hotspot, the frequency is lower than the control group that is no kinetochore proteins attached to the hotspot. So CTF lowers the crossing over frequency. IML, this kinetochore protein has no effect or no change on the frequency of crossing over unless you seriously look at the error bars that just extend a little bit, but practically no change between the control group is shown.
Okay, so moving on to D1, it is asking us to predict the effect on the number of copies of chromosome 8 likely to be present in the resulting daughter cells when CTF is attached to the hotspot. So we need to predict the effect on the number of chromosome 8 copies in daughter cells when CTF is attached to the hotspot. Our prediction is that CTF attached crossing over happens less frequently, which can cause problems with chromosome separation during meiosis. As a result, some daughter cells might end up with too many or too few copies of chromosome 8.
So question D2 is asking to provide reasoning to justify our prediction. So to justify our prediction, we say something along the lines of, for a proper crossover event to occur, homologous chromosomes during meiosis 1 are required for the chromosomes to align correctly at the metaphase. So if CTF prevents crossing over at hotspots, it may lead to non-disjunction, which is the failure of chromosome pairs to separate properly during anaphase, leaving some daughter cells with extra chromosomes and others with missing chromosomes.
Moving on to D3. D3 is asking us to explain how the presence of hotspots shown in figure 1 can increase the likelihood that a population will survive in the presence of selective pressures. Alright, for D3, we need to explain how the presence of hotspots could increase the likelihood that a population will survive selective pressures.
The presence of hotspots increased genetic variation by promoting more frequent crossing over and recombination in specific regions of chromosomes. This diversity allows a greater chance of beneficial genetic variation arising in the population. Population with more variation are better able to adapt to selective pressures and environmental changes, improving their chances of survival.
So that concludes our tutorial walkthrough of the 2024 AP Biology exam, long FRQ number 1.
We hope that you found these explanations and strategies helpful in helping you prepare for the biology exam.
Best of luck.